Hello everyone, I am Suchita Bharambe, Assistant Professor, Department of Microbiology, Government Institute of Science, Aurangabad. I am here to present my microteaching assignment for online refresher course in biological sciences conducted by UGC HRDC, Dr. Babasaheb Ambedkar, Marathwada University, Aurangabad. So today we are going to explore the fascinating world of gram staining. Objectives for the today's session are to understand principle behind gram staining technique, get acquainted with gram staining procedure accurately so that we can differentiate between gram positive and gram negative bacteria based on their appearance after staining. And finally, to interpret the result of gram staining to help in preliminary identification of bacterial species. So gram staining, it is widely used differential staining technique in microbiology, and it was discovered by Christian Gram in 1883 while working on the respiratory disease in a hospital of Berlin. The taxonomic importance of gram staining is in identification and classification of bacteria. It differentiates bacteria in two groups like gram positive bacteria and gram negative bacteria depending upon retention of primary stain crystal violet after treatment of decolorizer alcohol. Gram staining requires various reagents like primary stain, for example crystal violet, mordant like grams iodine, a decolorizer like alcohol, secondary stain like safranin. So the mechanism of gram staining is based on difference in cell wall composition. Gram negative bacteria have higher lipid content like 10 to 20% and lower peptidoglycan content like 5 to 10%. Hence, these gram negative bacteria after primary staining and mordant application, when they subjected to the application of decolorizing agent, which is an organic solvent, this organic solvent alcohol wash out the higher lipid content of the negative cell and increases the porosity of gram negative cell wall. Likewise, due to the lower peptidoglycan content, less shrinkage of the peptidoglycan pores are there. Ultimately, on the decolorization treatment, the gram-negative bacteria have increased porosity, which is responsible for washing out of the primary stain crystal violet, while gram-positive bacteria have lower lipid content and higher peptidoglycan content. So, after primary staining mordant application, when we add decolorizing agent, the organic solvent like alcohol, due to the lower lipid content, less increment in the pore size is carried out. Along with that, due to the higher peptidoglycan content in the gram positive cell wall, shrinkage of peptidoglycan pore is carried out more. Ultimately, the porosity of gram positive cell wall decreases on application of decolorizing agent and the crystal violet iodine complex retain inside the cell and the gram positive cell, even after decolorization, they appear violet in color. Okay. Now let's walk through the gram staining procedure stepwise manner. Simultaneously, we are going to discuss what is the staining status of bacterial cells in the smear at the end of that particular step. So we'll start with the first step, that is prepare smear using bacterial suspension on a clean grease-free slide. Air dry and heat fix the smear. So at the end of this step, all the cells present in the smear, they were colorless. After heat fixation of smear, cover the smear with primary stain crystal violet for 2 minutes. So at the end of this second step, all the cells in the smear, they were violet in color. Then wash the slide with gentle running tap water. Remember, the flow of water should be gentle. Harsh flow of water could lead to washing out of your smear. After that, cover the smear with mordant grams iodine for 1 minute. After this 1 minute, all the cells present on the smear, they were violet in color. Grams iodine forms a crystal violet iodine complex. Application of grams iodine form a complex with primary stain crystal violet. It is called as CVI complex, that is crystal violet iodine complex. After 1 minute of grams iodine, again we have to wash the slide with gentle running tap water. Then apply decolorizer alcohol in dropwise manner to the smear until the violet color stops to come out. At the end of this step, differentiation between the gram positive and gram negative started. As we explained in the mechanism, due to the higher peptidoglycan content and lower lipid content on application of decolorizing agent alcohol, the porosity of gram positive cell wall decreases and the CVI complex retain inside the gram positive cell and the gram positive cell appear violet in color. On the other hand, your gram negative cells, they have higher lipid content and lower peptidoglycan content. Hence, after decolorization treatment, the porosity of gram negative cell wall increases. It leads to washing out of the CVI complex. After decolorization treatment, again we have to wash the slide with gentle running tap water and cover the smear with secondary stain safranin for 5 minutes. At this stage, your gram positive cells, they appear violet in color. On the other hand, if gram negative cells are there, they get the stain safranin and appear pink in color. After 5-minute application of safranin, pour off the stain, wash the slide with gentle running tap water, blot dry the slide, air dry the stain smear and put a small drop of immersion oil on the smear and observe under oil immersion lens of microscope. The purple cells, they are the gram positive bacteria. The pink cells you observed, they are the gram negative bacteria. The results we can interpret like organisms which retain the violet color, they could be classified as gram positive bacteria, whereas organisms which retain pink in color, they are called as gram negative bacteria. Examples of gram negative and gram positive organisms we are going to discuss on this slide. So the gram positive bacteria belongs to the genus Bacillus, Clostridium, Corynebacterium, Streptococcus, Staphylococcus, Lactobacillus, Propionibacterium. On the other hand, gram-negative bacteria includes the genera Pseudomonas, Escherichia, Klebsiella, Acetobacter, Proteus, Shigella and Salmonella. These are the few examples of gram-positive and gram-negative organisms. Now I am open to any question you may have about gram staining and its significance in bacteriology. Please feel free to ask. So by the end of this presentation, you should be able to perform gram staining yourself and differentiate between the gram positive and gram negative bacteria using this technique. Now you know how to perform the gram staining. and how to differentiate gram-positive and gram-negative bacteria using gram staining technique. Here we are going to conduct a small test in the form of MCQs. The first MCQ is gram staining is based on difference in bacterial cells. A. Nucleus. B. Ribosome. C. Cell membrane. D. Cell wall. Yes, cell wall is the correct answer. Next question is, what is the primary purpose of gram staining in microbiology? And the option A is to determine the cell size, B to identify the presence of endospore, C to differentiate bacterial species based on the cell wall characteristics, and D to assess cell motility. Yes, C to differentiate bacterial species based on the cell wall characteristic is the correct answer. Next question is that after the primary stain, which reagent is used as a mordant in the gram staining procedure? Option A is crystal violet, option B safranin, option C iodine, option D alcohol acetone mixture. So what could be the correct answer? Yes, iodine is the correct answer. Next question. Which of the following bacterial species would typically be the gram-negative bacteria? Option A, Staphylococcus aureus. Option B, Escherichia coli. Option C, Streptococcus pyogenes. Option D is Bacillus subtilis. Yes, Escherichia coli is the correct answer. So thank you for joining me on this journey through gram staining. I hope you found this presentation informative and engaging. Have a good day.